Alright, so today we'll be talking about the immune system, and this will just be a general overview of what the immune system is, a very introductory lesson into immunology. There's a lot of topics here, so here's just the contents that we're going to be covering. We're going to start off with the two things which are just very basic things that we need to know for basically everything else. I'm assuming you have prerequisite in, let's say, proteins, basic cell anatomy like cell membrane, right, nucleus. That's about it. You don't really need to know that much else, just like basic high school stuff, right? So first we're going to talk about what antigens and antibodies are. We're going to go into MHC1, MHC2, and then we'll go from there and I'll explain everything else. Alright, so let's start with what antigens and antibodies are. So, antigens, you might have heard this before. These are just any proteins that trigger an immune response, okay? So you can imagine these can be found on bacteria, viruses, fungi, even non-living stuff like snake venom would be one, right? All these antigens do is that our bodies detect them and they think it's a harm, right? Sometimes they are, sometimes they're not. But either way, that's what the definition of antigens are. Now, antibodies are something different. These are any proteins that bind to antigens. Now, most of them look like this, right? With disulfide bonds here. You don't really need to know about that until later lectures. By the way, that's what antibody looks like, and what they have these binding sites. And these binding sites will bind to some protein, in this case, antigens, right? And these are all throughout your bloodstream, right, in your immune system, and they're also created by your immune system. We're going to go over how the process behind this, and in future lectures, I'll go over like how this happens exactly on a molecular level. But antibodies are extremely important, and they're also called immunoglobulins. You might see antibodies called like Ig something, like IgA, IgG. That's just they're antibodies, right? Because they just call them immunoglobulins because that's a bit more formal. But that's antigens and antibodies. Next, I want to introduce these things called MHC molecules. Now, MHC stands for Major Histocompatibility Complex, right? And basically, what this is, these are just a major class of molecules that are present on either cells or immune cells that are very important. And we're just going to cover the two primary types. And these two primary types are MHC class 1 and class 2. This on the right here, this is a class 1 molecule. In class 2, it's pretty similar. So like the alpha is like here, and alpha 2 is like here. And this is a bit different with like beta 1, beta 2. You don't have to know about that. We'll talk more about that in future videos. By the way, there are two classes. And we can think of MHC class 1 as almost like a... Oh, sorry, before I do that, just for a quick general knowledge, MHC just refers to like a gene locus on like in genes, right? And they code for MHC molecules. Just for your knowledge. It doesn't really matter that much, but either way, just for future knowledge. Either way, class 1 molecules, what are these? Well, these are molecules that are found on all nucleated cells, right? So this doesn't include red blood cells, but it does include, let's say, your skin cells, all your immune cells, right? Also include, let's say, your liver cells, heart cells, all of it, right? And what they do, you can almost think of them like a window into the protein activity in the cell. Basically, what it does is that a protein can bind up here, right? And that protein just basically tells any other cells what's exactly going on in the cell. Obviously, can't represent all the proteins, right? But for example, it might put like an antibody up here, and that antibody might signal that it's being infected, right? Or it could be like or just like a self identifying protein, right? And then cells will be like, okay, you're chill, you're fine, right? And this will be important very later on, and we'll explain why. The other class is called class 2. Imagine this is class 2. I think it's like B1, B2 here, or something like that. Imagine this is like a class 2 MHC protein. Now, these are only found on certain types of cells, and these cells we'll go over later. We call them antigen-presenting cells. Now, why do we call them that? Well, it's because these actually can contain extracellular antigens. So, for example, this might be an antigen from a bacteria, right? So it doesn't include like the intracellular proteins that the cell makes. These are proteins that it gets from the outside. So it could be bacteria, whatever, right? And we'll see why this is very important later. These are just like very basic things to know about the immune system. All right, let's move on to leukocytes. So these are the main stars of the immune system. And leukocytes, leuc just means white in Greek, and then cyte means cell. So leukocyte, white blood cells. There we go. So you've definitely heard of white blood cells before. And what these guys do is they fight infection. And so these guys are like the soldiers of your immune system, if you imagine like that. I'm going to use some soldier and military analogies because I think they work out pretty well. And then we have three types of leukocytes. And we have granulocytes, monocytes, and lymphocytes. What the heck are those? We're going to explain. All right? And these are the three types of leukocytes you need to know about. All right, let's start with granulocytes. So granulocytes, these are any cells that are filled with hydrolytic enzymes. So if we imagine like a granulocyte, right, here's our nucleus, they're filled with these little vesicles, right, that are filled packed to the brim with hydrolytic enzymes, right? And we'll go over, and these are all granules, all right? Granules. And we'll go over why they have this in future videos and how these are like, released and everything. But that's like generally what granulocytes are. Now if you imagine, all right, a lot of hydrolytic enzymes, right? Obviously, they're not trying to kill themselves. They're going to kill other things, right? And so we have a few types of granulocytes, right? And the first ones we're talking about are called mast cells and basophils. All right. For all intents and purposes, right now, we're going to assume they do essentially the same thing, except that mast cells they're found in tissues. All right. So if you imagine like your muscles, right? If you imagine your muscles and there's like some cavities in between the muscle, mast cells would be in there, right? So they're going to be like, as soon as the bacteria gets there, they're there, right? And then as basophils, they're in the blood. So if there's anything in the blood, they're taking care of it, all right? And so what do these cells do? All right, if we imagine like a mast cell here, right? And then let's say we have a basophil here. Basophils actually have like an S-shaped nucleus. We don't really have to, not very important right now. By the way, they have these little receptors, right? Every cell has receptors. And when they bind to a foreign antigen, right? Let's say there's a little bacteria, right? A little bacteria. And the bacteria has like an antigen, right? Let's say it binds to one of these receptors, okay? So these receptors are very specific, and they can bind to these antigens. What does it do? Well, remember, these are granulocytes, so they contain a bunch of granules. And so these granules get exocytosed, and they release a bunch of molecules. And what are these molecules? Well, I list a few here. They do produce a lot more, right? These are the main ones that I kind of want to talk about right now. And will come up later in future videos as well. First one is histamine. So histamine, this increases muscle permeability. So what does that even mean? Well, if you imagine a bunch of smooth cells, right? Smooth muscle cells, right? All right, so we got a little nucleus there. What histamine does, right? So some of these guys are histamine, right? They're going to cause the contraction, all right, of these muscle cells to get just a little smaller, all right? So they're going to contract, right? And now look, there's an empty space in between them. What does this empty space do? Well, a lot of cells can now travel through those muscle cells. That's what we call muscle permeability, right? And so we'll talk about this later, but like macrophages, neutrophils, they're all going to want to like climb through those muscle cells to get to where the action is, right? And some of these molecules actually attract them, so that ends up working out, right? The other one is heparin, and heparin will prevent blood clotting. So obviously you want blood to go fast and not slow with areas of infection, right? So you want to prevent blood clotting. Leukotrienes, these are very good for maintaining the inflammatory response, right? So inflammation, if you just imagine, inflammation is just where a lot of cell damage is, and so your body's like has a bunch of immune cells there to take care of it, right? That's what inflammation is. And also bronchoconstriction. So like shortness of breath, right? Pneumonia, stuff like that. Not always caused by bronchoconstriction, right? It could be fluid buildup and whatnot, but leukotrienes can cause bronchoconstriction, which you can imagine makes it harder to breathe, right? And then lastly, we have prostaglandins. Now, prostaglandins cause vasodilation, right? And vasodilation, if you imagine like a vein, right? What that does, or not a vein, blood vessel in general, it'll make it bigger. All right, that's what vasodilation does, and so more blood will be flowing, meaning more cells, more volume, right? So that's great for the immune response. And now prostaglandins also attract certain immune cells, and we're going to talk about what these immune cells are right now. So that's what mast cells and basophils are. Great, they release these molecules. Now I want to talk about eosinophils. All right, what the hell is eosinophil? Well, these are a huge class of like, okay, let's say like this is like a little basophil, right, or a little mast cell. Here it is, an eosinophil. All right, and these guys they actually have like a telephone-shaped nucleus, actually. So they actually look like they have sunglasses. So that looks pretty cool. And these guys, you can think of them like as like the big bosses of the immune cell. Not in terms of like leadership, but in terms of like these guys are they're strong. And they're larger than most immune cells, and they contain cationic peptides and major basic proteins. Don't even know exactly what those are. Just know they're very powerful. Imagine this guy's like a nuke. All right, this guy has a nuke that can like, like a radioactive sign there, right? This guy's a nuke, and what these guys do, right? Imagine there's like this little bacteria, right? They don't really care about bacteria that much, all right? They're more interested in the big fellas, right? Not allergies, sorry. Parasites. Imagine, I mean, you see, if you have ever seen like tapeworms before in videos, they're huge, right? They're huge, like you can actually see them in real life, right? They're like a little worm, right? So we need to deal with that somehow, right? Immune cells, like this little guy, like one of our cells might only be like this big to it, right? Like it's super small. But that's where eosinophils come in, because they have these nukes, right? Obviously, as in the form of granules, obviously, not that they don't actually have a full nuke, obviously. But these can do a lot of damage to even big organisms. And because of that, in very devastating allergic reactions, these guys actually drive a lot of those, right? If you think of anaphylactic shock and just very devastating impacts on allergies, a lot of it is caused by these eosinophils because, well, they're so powerful, right? A little basophil secreting histamines, I mean, they definitely don't help, but like that's not going to cause you to, like die from an allergic reaction, for example. Eosinophils, on the other hand, if they got nukes, and if those nukes can actually kill these parasites, yeah, I mean, they're going to cause a bunch of damage in the body. So that's what eosinophils are. You can think of them as like the big, like security, like bodyguards, right? Huge. And then now we want to talk about neutrophils. And now neutrophils are like the common soldier of the immune system. All right, they make up like almost around 2/3 of all leukocytes, right? And they're what we call phagocytes. Now, phag means eat in Greek, and cytes means cells. So they eat cells that eat, right? And so if we imagine like there's a neutrophil, right? They actually have like a I don't even know what the shape would be called, but like their nucleus kind of looks like that, right? And what they do, right? They actually do a lot of things. They're really cool. If you take a little bacteria, right, and it eats them, right? It eats them. We'll go over the phagocytotic process later on, but basically, okay, so he dissolves it with everything with like lysosomes and whatnot. There's a bunch of the antigens in him now, and he'll just release the antigens by exocytosis, right? And now antigens are in the bloodstream. We'll see why this is important later. I'll bring it up back like when we get to dendritic cells and whatnot. But okay, that's one thing it does. Other thing, let's say the bacteria is like really strong for some reason. All right. Let's say this bacteria is like it's just like really stubborn. Let's say it's a big bacteria too. Well, if it can't just dissolve them with lysosomes, right? It can actually convert oxygen gas into free radicals. And if you ever heard of free radicals in like health, right? They're very bad for your body. A lot of medicine is centered around reducing free radicals. Well, the reason is because free radicals are very deadly to living things in general, especially bacteria, but also the neutrophil, right? So what it ends up doing is it converts a lot of O2 gas, which obviously it has, being a live, aerobic cell, will convert it to free radicals and kill both it and the bacterium. And so this is what we call oxidative burst, because it basically sacrifices itself to get rid of an enemy, which is, it's very nice of him, right? That's another thing he does. Last thing he does that I want to mention, he does other things, but like these are the main things. He releases these things called nets, or neutrophil extracellular traps, right? And imagine, let's say we have a bunch of bacteria, right? Bunch of bacteria. And obviously he's outnumbered, right? What he can do, see this little chromatin of his in like this nucleus? Well, he can actually spit it out and form what we call nets, right? And these nets have histones on it and everything, right? And what this does is it immobilizes these bacteria or invaders or whatever they are, and localizes them to like a certain area, right? Because it's hard to move through these nets, and actually tags them for like enzyme breakdown or phagocytosis, right? So these are actually very cool because, I mean, to be fair, he dies in the process, right? But he spits out all of his genetic material to do so. So pretty cool guys. These are like your main troops of the immune system. Great. So they're phagocytes as well, right? So phagocytes, what exactly is that? Well, they're cells that eat, right? But they're not the main cells that also eat. You've probably heard of something like macrophages before, right? That's where we go to monocytes. Now, monocytes are both phagocytotic, right? But they're also called antigen-presenting cells. And what does this mean? Well, let's look at this macrophage first. All right, so we have this macrophage right here. All right, and let's say we have a little bacteria, right? And we'll actually go through the phagocytotic process. All right, so that's bacteria, right? He goes here, and then the macrophage pseudopods, get him, right? He engulfs him and everything, and then he engulfs him. He's in like a little endosome, right? And then this is the bacteria, right? And then what does he do now? I mean, there's lysosomes, right, containing a bunch of like digestive enzymes and whatnot. Fuses with the endosome, right? And then and then there's a lot of like, the bacteria breaks down because of that. Bunch of antigens in here now, right? Bunch of antigens. Now what? So now this macrophage, right? And macro, by the way, macro is Greek for big, phage, food, to eat, right? So these are big eaters. So what do they do with these antigens? Well, remember those MHC2 molecules I was talking about earlier? Well, these guys have a bunch of that. They're antigen-presenting cells, right? And so what they do is they put these antigens on the top of their MHC2. We'll see why this is important later, but this is what they do. And macrophages love to eat. These are like the main things. If you ever see a white blood cell chasing something to eat them, chances are it was a macrophage, right? Because they got pseudopods and everything to like extend. Very cool guys. Now, the other one's kind of freaky. This guy's called a dendritic cell. There we go. And he's also a type monocyte, so he can also eat bacteria, but not to a big extent. He more so loves eating antigens on its own. Remember when we talked about the neutrophil and how the neutrophil, once it engulfs the bacteria, breaks it down, it will release antigens into the bloodstream? Well, these guys will pick it up. All right. So let's say there's antigens in the bloodstream. They'll pick it up, determine if it's worthy enough to be looked in further, and it'll actually represent these antigens on its MHC2 molecules as well. So these are what monocytes do. They have phagocytotic properties, and they'll release the antigens on their MHC2 rather than just releasing into the bloodstream, right? Now, why is this important? Well, they're going to make their way into something called the lymph node, and they'll interact with lymphocytes. So, what exactly is that? Well, let me introduce them to you. Lymphocytes are found in the lymph nodes. So, what are lymph nodes? You can almost think of them as like military bases or garrisons of the immune system, right? And lymphocytes are just like the cells that kind of reside in there. And there's three main types. There's natural killer cells. All right, you might have heard of those before. There's T cells, and these guys will cover two versions. There's T cytotoxic cells, and then T helper cells. Then we have B cells, and in B cells we have plasma cells, and then we also have memory B cells. B cells. And we'll go over each of these in introductory detail, right? But these are basically lymphocytes, right? And I'll probably make a video dedicated solely to lymphocytes and their development as well. But right now, just know three types, right? First type, let's go over, is a natural killer cell. And what this guy does is, let's take a look at him, right? All right, there he is. And what this guy will do is, remember that MHC1 I was talking about at the beginning of this video? MHC1, which is basically the window into the cell, just think of it like that, right? Into the protein activity of the cell. Now, this guy is there to make sure, he's kind of like the police. All right, he's going out and looking for identifications. If you have an ID, you're fine, right? But if your ID is fraudulent, or if you don't have an ID, he's gonna kill you. All right, or if your ID has let's say there's a illegal immigrant stamp on it for some reason, well, he's gonna kill you. That's exactly the cases that we talked about here. no MHC1, right? So if you have no window into your cell, right? This guy's like, we can't have that. You have no ID. I'm gonna, kill you. Mutated MHIC, right? Sometimes there can be mutations in the proteins of it, right? And that can be indicative of cancer cells, right? Could also just be like, it's just not good for the cell either way. So he's there, quality control, kill. And sometimes there'll also be an IgG antibody, right? Remember IgG? Remember we talking about IG stands for immunoglobulin, right? Immunoglobulin. That's just another word for antibody, right? And if there's an antibody on this MHC1 molecule, well, he's going to kill him, all right? So how does he do this? Well, he does this via apoptosis. And apoptosis, if you're not familiar, is programmed cell death. So how does he do this exactly? Well, he releases these things called granzymes, right? Let's imagine we have there's this guy, and we have a cell. All right, and he has no MHC1. Or let's say he has a mutated MHC1. All right, boom, very mutated, right? He's going to release granzymes, right? Let's say the granzymes. And these granzymes will move into the cell and actually activate pro-apoptotic genes, right? And so if you imagine the genes of a cell, there's genes specific for the initiation of apoptosis, and he initiates that, and this cell ends up dying. Great. So these guys are basically, just imagine as police, right? They're checking for your ID. You have an ID, it's not fraudulent. You haven't been tagged as like a wanted criminal. You're fine. Okay, now let's go to the T cells. Now T cells are a type of lymphocyte, right? And they have these things called T cell receptors. Shocker, right? But these are very important. These are always specialized for a certain protein or antigen to bind to, right? So this is where our antigen-presenting cells come in. Because remember the macrophages and dendritic cells, they had the MHC2 molecules with a little antigen on it, right? Well, they're going to try to find, let's look at the macrophage, right? And he has this little antigen he wants to give this one. Now, there'll be a bunch of T cells, right? A bunch of T cells. And each one of these T cell receptors are different. And he's going to find one that matches. Once he finds one that matches, let's say he matches to this receptor, right? He's going to tell this T cell to multiply and differentiate into two types, all right? We're going to go over how, like, the nitty-gritty of, like, which type it differentiates into later on in different videos, right? But just know it can differentiate two types. First one is a cytotoxic T cytotoxic cell, also known as killer T cells. So you've probably heard of that term before. And what this guy does, I remember he has these T cell receptors, right? He's going to go out into the body and look for cells whose MHC1 molecules have this antigen, and then do the exact same thing our natural killer cell did before, right? Which is do apoptosis. But you see how this is a little different from natural killer cells, right? Because natural killer cells were only looking for like the presence of MHC1, if it's mutated, if there's an antibody on there, right? These guys are looking specifically for the antigen, right? So you can almost think of these guys as like assassins. All right, they have a certain like a list, a wanted list, and they're looking for that specifically. And if everyone else, right, they don't really care about them. So they're like the special agents looking for specific, maybe a bit more elusive cells. And usually they only target cells, right? So this is a very big role in cancer, right? Cancer is a big one, or viral infected cells, right? Bacterial like infected cells, if that happens, right? That's sort of what T cytotoxic cells do, or killer T cells. The other one that is differentiated to is called a T helper cell. So if you imagine what they look like, kind of look like this, right? And what do T helper cells do? Well, they activate B cells. So what exactly is a B cell? Okay, well, before we move on, remember these guys have one type of T cell receptor, which binds to this antigen that this macrophage gave. Just remember that, okay? B cells. So these are another type of lymphocytes that are found in common lymphoid organs. So this can be like the spleen, right? Or it can be the lymph nodes. I'll go over like this nice lymphatic system in a different video. But just know, like, military bases of the immune system, right? A lot of them, this is where immune cells like gather, exchange information, plan out, strategic, maybe not that advanced, but like this is where a bunch of immune cells gather, right? And they're all over your body. By the way, I digress. These B cells, right? Look at these B cells. They're all naive. They're just chilling, right? But they have these things called B cell receptors. Now these are very similar to T cell receptors in the fact that they have specific binding sites for proteins, for antigens. And now let's look at this helper T cell, right? Helper T cell has their T receptor that's specific for a specific antigen, right? Now he's gonna find the one that matches. Okay, this guy doesn't match, and let's say he finds the one that matches. This guy, right? Now what does he do? Well, he activates them, and when he activates them, this guy undergoes clonal propagation. Okay, that just means like he starts multiplying. Like a lot of them, and then now they differentiate into two types of B cells. We have one is called the plasma cell, and the other one is called the memory B cell. And now the plasma cell, okay, let's say this guy differentiates, right, into a plasma cell. What does he do? Well, he has B cell receptors for, what's it called? Or sorry, he knows exactly what antigen he got, he ended up using, right? Now, that antigen can have a specific antibody associated with it, right? That's what this plasma cell does. He starts producing a lot of antibodies, right? That will bind to the antigen that this T helper cell was originally primed for by the macrophage. Remember, the macrophage, right, presented this antigen, right? And then now we see this beautiful cycle where we end up with a plasma cell, where he secretes these antibodies to bind to these antigens, okay? We'll talk about what these antibodies actually do later on, right? And we also have some memory B cells. So this also happens. And what do memory B cells do? Well, they're kind of just chill there. I mean, bro's just chilling with his receptors and all, but he's already primed with this antigen. He already knows this is the antigen he needs to use. And when you encounter this antigen again, let's say after this first infection, and it comes back, this guy becomes a plasma cell really, really fast and starts producing these antibodies. That's why the second time you get infected with the same infection, right, is actually a lot faster, or you might not even notice you're sick, right? That's why you can be safe to say, like, okay, I've been infected already, doesn't matter if I get infected again. That's because we have these memory B cells. B cells. Okay. Now these antibodies. What the hell do these antibodies actually do? We've talked a lot about them. They're very important somehow, but why? That's where we go to humoral immunity, right? And we want to talk about a specific part of it called the complement system. Now, the complement system is a system filled with complement proteins, all right? So we have complement proteins like C1, C2, all the way to C9, all right? And there's three pathways that the complement system falls under. And the one involving antibodies is what we call the classical pathway. Now, I'll explain what this is. Imagine we have a little bacteria, and this bacteria has a little antigen, right? Now, what happens? Remember, we had the plasma cells, right? This is the plasma cell, and he's secreting the antibody for this antigen, let's say, right? Well, this antibody will bind, right? And let's see what happens. Well, we had C1, right? Remember C1 up here? Will actually bind to this antibody. And then we have C4 that binds to this. Then we have C2, C3, C3, C5, C6, all the way to C9. Okay? We're talking about what this does actually does later and more in depth in other videos. This is what happens in the classical pathway. Just know this for now. Alright, bear with me. Alternative pathway, what happens actually is that we skip the antibody step, and just so happens that C3, right? This is C3, binds to this bacteria, right? And imagine what happens after C3. Well, C5 binds, right? Then C6, C7, all the way to C9. Okay? Alternative pathway. Now, the lectin pathway. Let's say we have a bacteria, and he's covered with something we call mannose. All right, mannose. Let's write that, mannose. Now, our bodies have something called mannose-binding lectin. Let's take this right here. This is mannose binding lectin. Now, what does this do? Well, mannose binding lectin, C4 will bind to mannose binding lectin. Right? Now, imagine what happens after C4. Well, C2, C3, C5, C6, all the way to C9. Okay? Those are the three pathways of the complement system. And keep in mind that antibodies are only directly involved in this one. Okay? The classical pathway. Now, what are the importance of these complement proteins? Why do we care that they're on these bacterial cells, right? Or whatever, or fungi or virals, right? Well, these let's imagine the bacteria again. These proteins will break off and form something that we call the membrane attack complex. We'll go over this much more in depth in future videos, but either way, this is the basic function of it. This bacteria, right, with the antigen and the antibody and it blah 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 forms a membrane attack complex. Now, what does it do? Before, the bacteria is perfectly fine, right? He's just chilling. The bacterial wall, the cell wall is very strong. Like he's chilling. But what this guy actually does, the membrane attack complex, will actually insert itself into the bacteria. So now there's like a hole in the bacteria, right? And what flows through this hole? Well, water can. And small ions, but mostly water, okay? Now imagine the concentrations of proteins or ions in a bacteria versus the blood vessel or the tissue extracellular space. Obviously, the concentration in the bacteria is going to be a lot higher, right? If let's say bacteria concentration is like 50, right? Outside might only be like 10, right? This should make sense. Because bacteria has a lot of stuff going on. Everything has to be controlled, right? There's the reason the cell wall is so strong is to prevent environmental challenges into the cell. So when you now add this membrane attack complex, what will flow through? Well, think about osmosis, right? Water is going to go to area of higher concentration, right? So water flows into the cell. This happens really fast. A lot of water all at once into the cell, and the cell blows up. Lysis, right? This should make sense if you imagine, it's like a very small cell, huge blood vessel. They can't compare the size in, right? Blood vessel, the blood obviously doesn't have that many proteins and ions compared to the small little concentrated bacteria, so a lot of water rushes in and it bursts, right? And that's how antibodies can end up causing lysis, and that ends up killing the bacteria, which is very good for us, right? Another thing the complement system does, right? This C3 can sometimes induce macrophage phagocytosis, so it makes macrophages want to eat more. But this is what it's mostly about. So that's the complement system, okay? Alright, now I want to talk about the immune response. So there's two types. There's the primary and the secondary. Primary is the first exposure to an antigen. So this also can kind of act like a recap. I'm just going to go through the entire pathway so far, again, from the emergence of, let's say, a bacterium. Alright, there's a bacteria. What happens first? Well, remember, we have our mast cells or basophils, right? That will end up binding to this antigen, right? And be like, hold up, that's not meant to be there, right? I'm going to release a bunch of molecules. What do we have in these molecules? We have histamine, right, which makes muscle more permeable for macrophages or neutrophils, right, so they can get in there and eat this guy, right? Heparins are another one, increases blood flow, right? Leukotrienes is another one, bronchoconstriction and inflammation, like increased inflammation, right? And also prostaglandins, right, which can attract other immune cells, and also vasodilation, so there's more blood in the system, right? So let's say he does a good job and a macrophage comes in, right? Macrophage. And macrophage will eat him, and let's say he eats him, goes in there, and then he represents the antigen on his MHC2 molecule, on his MHC2 molecule. Alternatively, the neutrophil, remember these neutrophils, right, can also eat him, right, and then secrete his antigens. And then dendritic cells, right, dendritic cells, right, will take those antigens and then represent them on their MHC2 molecules. So those are two possibilities. Now, what do these guys do? These guys go to the lymph nodes. Imagine this is a lymph node. Lymph node. Now, what are in here? There are T cells, naive T cells, and naive B cells. So we got a little macrophage, right? He wants to come say hi, introduce himself to the T cell. He finds the right one that has the right receptor. Let's say this guy has the right receptor. Now this guy is going to turn into a C cytotoxic cell, right? That will go out and try to find cells that express this antigen, or a T helper cell. And this T helper cell, which knows what like antigen this was, right, will go and find the corresponding naive B cell. And remember, the B cells have the B cell receptors, right? And when it finds the right one, let's say it finds this right one, right? Their receptors are gonna like match. It's like, oh my god, we're soulmates, right? Let's go. He's gonna cause him to clonally propagate, right, or clonally expand, and he'll produce two types of B cells. We got plasma cells, right? And we have memory. Plasma secretes antibodies, right? A bunch of antibodies, right? And then the antibodies, let's see, there's another bacteria here, right? With the same antigen. Antibody binds on, and then you have your whole pathway, C1, C4, right? C2, blah, 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 all the way to C9, forms a membrane attack complex, right? Membrane attack complex, water goes in, lysis, boom. Alright? That's humoral immunity. Remember? This is a very, very fast recap. It's okay if, it's too fast. What's important is that we formed a memory B cell, right? That has these receptors for that specific antigen. That is very important. Now this memory B cell is just going to be chilling out in the bloodstream, just circulating. He's just chilling. Alright? Now imagine we get exposed to the same antigen again, right? Say this bacteria wants, it's round 2, right? And this is the exact same antigen. Well, eventually, right, we're going to get this memory B cell with this B cell receptor, and he's going to be like, hold up, I've seen this before. And he's going to very quickly, just within a few days, turn into a plasma cell again to secrete antibodies that will now bind to these bacteria, right? Bind to these bacteria, form membrane attack complexes, lysis, and also attract other immune cells to attack these bacteria, right? That happens really fast. And so we don't even remember, or sometimes we don't even notice the second exposure to an antigen because our body is so efficient at it. Because these memory B cells I guess this would be the memory B cell, turns into a plasma cell really fast because it doesn't have to go through the T cell process and all that. It can and it might, but you don't have to go through all of that, right? And so it actually ends up being really fast. This is a secondary immune response. So that's why when you're sick with one virus, if you get infected with the same virus again, it's not going to really do anything because your body kind of already knows what to do. And usually these antibodies, right, also circulate in your bloodstream for a while. And so even if it comes again, these antibodies kind of just immediately just attack and, dissolve everything, right? So that's what the secondary immune response does. And speaking of secondary immune response, we can kind of see parallels into immunity, right? This is the whole point of what immunity is that we actually are able to do these things really fast. So that's where we get to immunity. So we have two types of immunity. We have passive and active, all right? Passive is when there's no immune response that's necessary. So this is very immediate. So if you imagine natural immunity, when you're a baby and let's say you're breastfed by your mother, the breast milk actually contains a lot of the mother's antibodies, and that will be passed into the baby through the breast milk. And so that's passive immunity because the baby didn't do anything to get those antibodies, right? It was just kind of chilling there. Boom, antibodies in the blood. Great. I have a bit of advantage in case anything, any microbes try to attack. My antibodies will help me out. The other one is artificial. So let's say you are bitten by a snake and there's some snake venom, right? You can also have antibodies injected into you, which is antivenom, right? And that will help your immune system handle things a lot better. Now I want to go into active immunity. So active immunity actually requires an immune response. So this actually takes time, right? And so naturally, this is kind of just like you're infected with a virus or a pathogen. Your body takes care of it, forms memory B cells, right? Just a normal way. But sometimes it can be dangerous, right? Imagine a deadly virus or a deadly pathogen like malaria or COVID, right? And it might be a little dangerous to go through that route, so that's why we have invented artificial active immunity. Might have heard of vaccines, for one, right? Vaccines. Now, what they do is instead of giving you the whole shebang, right, the whole virus in its pure form, right? Let's say you have a little virus, right, with covered in antigens, right? Well, that would be kind of be anti-intuitive, right? So instead, they kind of either kill this virus or make him really, really weak, so where he can't really do much harm, right? And what you do is you still tell your immune system this guy's an invader, but he might not be very strong, but your immune system still takes care of him anyway. Let's see injection into your bloodstream, right? So there's a bunch of these viral particles with antigens in your bloodstream, and they get taken care of by your immune system, and they form memory B cells and everything because they're like, this is this is not good. That's why sometimes you get side effects during vaccines, right? You might get a fever, like the COVID vaccine. A lot of people get fevers. I got a fever after the COVID vaccine, but it was worth it because it formed the memory B cell, right? And so when I actually got COVID, my symptoms were around the same as the actual symptoms I got taking the vaccine, right? Which was just like a very mild fever. So that's like the beautiful thing about vaccines, that we can kind of almost imitate like actual infection by not actually damaging our body at all. Because I mean, these viruses aren't going to do much. Like maybe they might infect a few cells, but like not at all, right? So that's kind of what immunity is. But I also want to talk about allergies and autoimmune disorders because these are also a very important part of immunology. And so if you think about these antigens, right, how does your body know what antigen is like bad and good? Because think about it, we have like good bacteria in our body all the time, right? We have our own cells, right? They also have antigens. We eat food, like food can have antigens on it too, right? So how does our body tell which ones are harmful and which ones aren't? And this research is actually not as like what's it called? There's not that much research for this, or there is, but like there's not a lot of conclusive theories. But some leading theories is that our body knows what is harmful and what is benign, not because it just knows, but by association. Like imagine you get infected by a virus, and there's a lot of like localized like inflammation and everything. Chances are it was caused by that virus, so your body's gonna be like, oh, that antigen is associated with damage, so clearly that antigen must be bad. We're gonna go attack that. Whereas let's say your gut bacteria, right? They're just chilling. They have antigens, but there's no harm being done. So your immune system's kind of like, well, they're chilling. You know, chances are we don't need them. We don't need to take care of that. And so that's why we can like actually eat food without like blowing up each time. But some people are like that, and that's what we call autoimmune disorders or allergies, like the more common type, right? And so why do sometimes we develop allergies as kids or as adults, right? Well, there's a theory. I think the earliest study I found was like in like the 1970s or something like that. Is that every time there's a pathogen, let's say, that infects our body, let's say it's a virus or like the flu, a lot of times during these times, it's pollen season. It's spring or whatever, and there's a lot of pollen in the air. And over time, your body associates. Well, I got sick during pollen season. There's a lot of pollen in like the nasal area, right? And I'm sick from a virus that is mostly, respiratory, right? Therefore, the pollen must have caused it too, right? Obviously, it knows the virus caused it as well, but if the pollen was there, why take any chances? So then, that's the theory is you would develop an allergy to that pollen by association of it, like being associated with the virus, right? Um, it's not like super like I don't think the sample size was super big for the article I read about this. I think it was only like maybe a few dozen kids, right? Um, but this is a leading theory for why this happens. And I'll go over this more in like future videos about allergies and autoimmune disorders, right? But this is a leading theory. And obviously genetics play a big role, right? Because some people develop them as kids, right? Some people develop them as adults, some people never develop them, right? So it's very dependent on genetics. But the leading theory is that's sort of why that happens. We also have autoimmune disorders, right? Where the immune system gets confused and thinks our own cells are bad and will conduct, the whole immune response we just covered, but instead of on a bacteria, it's on us, right? And if you imagine our eosinophils, right? These guys are specialized for bigger cells, like parasites and whatnot, right? Remember the nuke, right? He has a nuke. So not bacteria. All of our cells are bigger than bacteria. This doesn't get called in, right? And he's gonna do some damage. So that's sort of a really scary impact on, like, let's say allergies. Like, let's say you have a big, like, it's not just like small pollen grains, right? Let's say it's something bigger. Let's say you ate a food like peanuts or something, right? That's a lot of mass. And so this guy can get called in, and then he does a lot of damage, a lot of inflammation all over the body, and that's what anaphylactic shock ends up, being, right? I'm not very well versed in allergies and the processes, right? But I'll make a video on that when I'm more informed. But that's generally what happens, is that your body, thinks this is a bad thing, calls in the reinforcements for something that's actually harmless, or even our own cells, and then that's where autoimmune disorders come from. And a lot of people with autoimmune disorders, they're on immunosuppressants. But that's also a bad idea, because what if there's an actual infection, right? But the immune system's like, too weak because, you've been taking medication. And so like a simple flu that we can recover from in like a week, these people can actually like get really hurt from. So it's a very tough thing. Doctors are still trying to figure out to solve properly, because the immune system's so complex and the system is so perfectly imperfect that these things can happen, right? Um, but yeah, that was a big overview of the entire immune system. There's still stuff that I left out just for time's sake, and I'm also not very informed in a lot of topics, so I kept it more introductory. Um, but let's do a quick recap, right? Quick recap: antigens and antibodies. Remember, antigens, any proteins that will induce a immune response, right? We got antibodies that bind to those antigens, right? We also think about MHC1 and 2, right? Remember that MHC1 is like a window into the protein activity of the cell. MHC2 is like on the antigen presenting cells, right? It's like, oh, look what I found, mom, right? They present the little antigen that they either like they found in the bloodstream, or the macrophage intook, broke the bacteria down and presented as antigen, right? That's what MHC1 and 2 do. We covered granulocytes, right? Remember we had mast cells and basophils. What do these guys do? Well, let me use my pointer. What do these guys do? Well, they release a bunch of molecules upon finding a foreign antigen. They're like, this is not meant to be here. I'm gonna go alert the immune system. They release a bunch of molecules, including like histamines, right, heparins, right, leukotrienes, prostaglandins, right, all of that good stuff, to attract other immune cells. We also had eosinophils, right? Remember, these are the big dogs, like the sunglasses, right? Sunglass nuclei, right? And they have like nukes, right, in them that are very, very deadly to especially like parasites or even our own cells in some cases, right? So they're very important for like very big invaders that are not quite just like viruses and bacteria, right? What's that neutrophils? Remember, neutrophils are really cool. They compose most of the white blood cells. They can release nets, right? Neutrophile extracellular traps, right? To tag it, like let's say you have a bunch of like bacteria there, like a big trap with histones and everything, right? Um, they're very important. Also, oxidative burst, right? If there's a very strong bacteria that's more resistant to like the lysosome, right? They might convert O2 into free radicals, and that can kill both the neutrophil and the bacteria. So almost like a suicide bomber, right? So they're very, very important. Also phagocytotic, right? So they'll actually eat, pathogens, just like monocytes will. And monocytes are also antigen-presenting cells, remember? Especially dendritic cells. They specialize in that. And so what do? Like macrophages and dendritic cells, when they present them to lymphocytes, right? Primarily T cells, right? We also have natural killer cells, which, remember what these guys do. They look for, like the police looking for identifiers, right? If you have an ID, the MHC1, right? If you have an MHC1, you're fine, but if it's mutated or if it has an antibody on it, no go. You're getting detained, which is, code for apoptosis, right? But I digress. These antigen-presenting cells will present them to T cells, right? Remember T cells? And T cell have these T cell receptors that will bind to that like have unique binding sites, right? And so once the antigen-presenting cells find the right T cell, they're gonna differentiate into both T helper and T cytotoxic cells. Remember, these are also called killer T cells. And so the killer T cell will go out and look for that specific antigen, right? These are like the assassins, right? They have a list of like people they need to or cells they gotta kill, right? But the other one is a T helper cell, which knows the antigen that was just given, right? And he will activate B cells, right? Remember, he'll activate B cells. They connect. He'll find the right B cell, the right B cell receptor. They'll meet, and then he'll cause the B cell to proliferate and differentiate into memory B cells and plasma cells. Remember, memory B cells, they act as like memory, like they'll be chilling in the bloodstream, ready for the next infection if it comes. Plasma cells, on the other hand, will release antibodies. And what do antibodies do? Well, the antibodies that will bind to the antigen that the antigen presenting cells originally gave the T cell, which then gave the B cell. Remember, right? That antigen is part of the classical pathway of the complement system, and that can induce a membrane attack complex that inserts itself into the bacteria cell wall, induces lysis by putting a lot of water to flow into the cell because the cell innards are a lot more concentrated than the outside blood plasma, right? And there's also two other pathways in the complement system. Remember the alternative pathway where C3 just binds to the antigen itself and then the rest follow, or the lectin pathway where you have bacteria with mannose in the cell wall, and our mannose binding lectin, which is present in our bodies, will bind to that mannose, right? And then all the other the complement proteins will bind and also form the membrane attack complex. All right. We also talked about immune response, both primary and secondary. Remember, primary is the first exposure, which you have to go through like the whole thing, right? You have to go through like, okay, you got to activate the T cells, but activate the B cells and whatnot. But there's also the secondary one where the memory B cell will just immediately turn to the plasma cell, skipping all of those steps and immediately just producing antibodies, which is a lot faster. Remember? We also talked about immunity because of that, right? Remember, we have passive and active. Passive, you didn't have to do anything for it. And you can also do it in active where your immune system actually has to work for it to build those memory B cells. And that can either be natural or artificial. So natural, which is either like from your mom, right, drinking her breast milk, or it could be just from like actually exposure to the pathogen, or it could be artificial. Maybe snake venom, and so you got to inject some antivenom into yourself, right? That's passive. Or it can be a vaccine, right, where your body cells will work to make those memory B cells. And we also talked briefly about allergies and why our body can, like theories, right? But why our body might, I'd look at benign substances and be like, that's a problem. I'm gonna go be inflamed because of that, right? Again, this is just a very broad overview, and it's a lot of information condensed in, I don't know how long I've been recording, but probably not that long anyway. I'll be going over basically every single one of these topics in more depth in the future and in future videos. But it is a lot, and this is very introductory. Like, I didn't even go through any of like the physiological pathways really at all, or any development of these cells. There's a lot of branches that like branch into hematology as well, so there's like a lot of, a lot of information that's missing. But hopefully this will give you like a nice overview of what the immune system is, gave you some like just a good understanding and appreciation for the complexity of it, right? This only touches the surface of to be fair, it touches the surface of a lot of different topics, but there's a lot more to it than just, T cell activates B cell, right? There's a lot of interactions that I left out. One, because I'm not super well versed in them anyway, right? I need to do a lot more research and compile a lot more information to confidently give those videos now. But also, it would just be too much for an introductory video, right? But I hope this helped if you are either reviewing or if you are just trying to learn or get a different perspective, right? We covered all these topics. Hopefully they made sense. And yeah, stay tuned for more videos on these topics. I'll be talking about granulocytes in depth, or monocytes as well, or development of lymphocytes, everything, right? Or the specific, activation of the complement system proteins. Oh, um, but yeah, this was just an introduction. Yeah, hopefully it helped.